started. I will return the exam after class. Hopefully you guys can see this on Brightspace. Apparently not still the quiz. Apologies about that. New system still working. But I will try to get some results as well. But pretty much everybody's ability to know what the solution. I'll post a couple of critical details about the exam, some areas where we focus on things that are sectional. Put that on Brightspace as well. And if you have any questions, office hours. Right? Okay, so let's pick up where we are. We've made some RNA, and pretty much there's some notable exceptions. But for the most part, in a prokaryote, there's not much more that has to happen to that mRNA. That's not really true in the eukaryotes. So what we'll talk about in the next couple slides anyway is purely eukaryotic in nature. We pointed out that at the 5' end, there was the sequence that is not strictly speaking coded for in the RNA, but that gets added automatically, called the 5' cap. It has this structure that I'm showing you here. This isn't the entirety of it, of course, it's attached to the entire RNA. We talked about the process by which that gets put together, and so that's obviously very important. We noted that there's a couple of critical features of this 5' cap structure that we should mention. Of course, knowing the structure is really important, but really to focus right on the green box in the center, we see a very unusual triphosphate linkage that took quite some effort to make. There's really only one nuclease that can break that. So one important role of the 5' cap is just to keep the 5' end from being degraded by any exonucleases that happen to come along. RNA is very, very labile, easy to destroy. This isn't just necessarily the triphosphates, however. By methylating the hydroxyl groups, the open hydroxyl groups on the sugar riboses, that also increases the stability of the 5' end of the RNA, making it less likely that it will be degraded. There are other important aspects of the 5' cap as well. We haven't really looked at what they are, but just to kind of jump ahead a little bit. An mRNA will not be translated into protein. That's the case. Most mRNAs anyway, if it does not have a 5' cap in the eukaryotic system. We'll see why that is in a later lecture, but for now you should know that. In fact, an mRNA will not actually get to leave the nucleus into the cytosol if it doesn't have a 5' cap. Not having a 5' cap is a pretty good signal that there's something wrong with that mRNA, and maybe it'd be better to just chop it up into pieces instead of trying to translate it. So we need it for translation, we need it for export from the nucleus, and we need it to protect the growing and then fully made mRNA. Again, no organelles have caps. We talked about cap snatching, so here's a couple details on a cap here, including the methyl donor that we'll see again. What about the other side? So now we make our RNA, we make our RNA, we make our RNA. We noted that POL2, which is the polymerase that's making our mRNA, does not, strictly speaking, have a termination sequence, at least nothing that's going to make the RNA polymerase fall off. And that's because it really kind of doesn't matter. As long as it makes it to the end of the coded sequence, and a little bit beyond that, that is a pre-mRNA that is capable of eventually being translated. And the reason why is we're going to make some modifications at that 3' end of the mRNA. Anyway, specifically speaking, we're going to add just a series of A records. Just A. And that could be generally roughly 50, as many as 250, sometimes more. Functionally speaking, again, you don't get to leave the nucleus without it. You won't be translated without it. So most importantly, it's just not really any interesting information. So if you want to lose a couple of nucleases, that's really not so much of a big deal. So it's really serving at the 3' end many of the same functions that the cap is serving at the 5' end. Protection, enabling translation, and enabling proper escape from the nucleus. You're not going out without it. The key is that here there is some heterogeneity actually with the tail, the poly A tail, which is what we call it, is actually getting made. The process itself is pretty much uniform for all RNAs, but it's not necessarily really a precise process. That is, there isn't this one discrete point where the tail is just going to be added and it's some sort of a clear signal, add the tail exactly here. Instead, there really are two signals. One of them is 5' where the tail is going to be added, and that is that sequence over here. So that's in the nucleus. About 10 to 30 nucleotides repeated that particular sequence there. And then on the other side, what we call the 3' sequence is this GU-ish sequence of 20 to 40 nucleotides. A lot of variation in the sequence, so there's not really much point worrying about exactly what it is, just that it's a very G-rich sequence. And then somewhere in between there, an endonuclease is just going to cut. And remember, anything that the RNA polymerase II added after that point is just gone, right? So again, that's why it didn't really matter where we terminate, because we're going to cut off anyway after those two sequences. At this point in time, we have a free hydroxyl. So a very unusual RNA polymerase is going to come in. It doesn't require a template, unlike the other RNA polymerases that we've seen. And it's just going to start adding A's. Just A. It's going to do so using the same chemical mechanism we've seen. So energetically, this is going to be driven forward, of course, by the loss of the pyrophosphate and the reconstruction of the pyrophosphate. No template is required. And that's just going to kind of go on for a while until it doesn't really. And so we'll end up with a poly A tail that's generally anywhere between 50 and 250 A's all the way at the end. Can be a little longer, definitely could be shorter. We'll see that keeping it shorter is sometimes a regulatory mechanism. And then this will get coated with a protein that's not shown here, called the poly A binding protein. It basically coats the tail. We'll see that that's actually a pretty important protein as well. But for now, its real job is going to be kind of helping with the poly A tail. We'll see it does have a very important function in the way that the tail actually works. And so, yeah, that's really the major processing on the ends. We do have an additional processing step that is necessary for most mRNAs. There are some exceptions to this, but most mRNAs are not made in the form where they translated. That is, there are sequences within those mRNAs, within the bulk of the mRNA itself, called introns. I'm sure you've heard about these, that do not, in fact, or should not, in fact, be translated later on when this is used to make protein. And so these intron sequences, which are between the exon sequences, those are the ones that do have information, have to be removed. Okay, so what do we have here so far? We've made mature mRNA, we've made capsid, we've tailed it, but we need to remove those intervening sequences, because those sequences, although they are there, and they are presumably important that they are there, are not meant to be translated eventually into protein, right? So it's not the information that they actually carry. How do we know, in fact, that this is the case, and that in fact, in many cases, mRNA genes are much, much, much larger than the final amount of information content necessary to make the protein? There's an experiment. We did this, I think, briefly in the first lecture, so we don't need to talk about it too much. Essentially, it's this. If you take a gene, a gene segment, and make an mRNA out of it, right? So you have a piece of DNA that you know has a specific gene associated with it, and you go and you translate it, and a mature mRNA is made. You can then, under certain experimental conditions, cause that mRNA to stick to the original DNA from whence its information was derived by Watson-Crick base pairing, right? So this requires you to take your original piece of DNA, know that it has a specific amount of genetic information, allow that DNA to be used to make a mature mRNA, and then stick the mRNA effectively back on the original DNA. And if you do so, you'll notice something interesting. You would expect if all of that piece of DNA had been used to make mRNA, then they should perfectly hybridize, right? They should basically have all of the mRNA hybridizing or Watson-Crick base pairing onto the DNA. That's what you would expect. But what we see instead are these little loop structures of DNA that seem to keep coming out of it. In other words, a portion of the mRNA is sticking to the DNA, and then there's a bit of DNA that's not sticking to any mRNA. And then for a little while longer, there's some DNA and mRNA that are stuck together, again by Watson-Crick base pairing, and then another long segment of DNA that is not in the mRNA. And these structures called R loops were the first evidence that all of the information that is present in the gene originally in the DNA is not ending up inside of the final mRNA that's being used to make protein. And in some cases, those introns can be fairly modest. In other cases, you can see here, for example, with the chicken ovalbumin all the way at the bottom, there is far more sequence present in the intron than there are in the exons, and the exons, again, are the sequences that are being used to code up the final protein. So most, in some cases, of the gene never ends up, a protein that, coding gene, I should say, ends up actually being used to make protein, at least not directly. So that was the first evidence that we know that, in fact, there are these things called introns. So it turns out that upon chemical investigation of this, There are really 4 different types of introns or 4 different splicing mechanisms. We're going to focus on them biologically, more so than chemically, but they do have different origins. One of them is, well, they're just named pretty easily, actually. The group 1 and the group 2 introns. These are both self-splicing in the sense that means that no outside help or assistance or energy is required. So if we're looking at the group 1 and the group 2 introns, you don't need anything else. In other words, under the right conditions, when this mRNA gets made, the splicing reactions will happen. So we call that the self-splicing introns. There is also another type of intron here called spliceosomal intron, but also known as nuclear splicing. This is what's actually happening for your introns, which is why you should care about it. This actually requires an enzyme known as the spliceosome. Now, just to be clear, when we call it an enzyme known as the spliceosome, this is not like an enzyme like chymotrypsin or trypsin from last semester, where there's a single polypeptide chain. This is an enzyme that is massive, requires hundreds of polypeptides and nucleic acids, in fact, to do the actual splicing process. So I'm just going to call the spliceosome an enzyme, but just keep in mind, this is a very complex multi-subunit enzyme that contains both protein and RNA components, as well as some small molecules and ions. But in this case, these are introns. Spliceosomal introns will not remove themselves, unlike group 1 and group 2, which we'll start looking at. And then we have tRNA introns, and these are the wackiest type of introns in the sense that they use very different chemistry than the group 1, group 2, and spliceosomal introns. But fortunately, we've actually seen all that wacky chemistry before. There's actually a special nuclease to do the cutting, and then a ligase that's very much like DNA ligase in terms of its final mechanism. So even though they are the most exotic, and again, I don't know why nature went so exotic with tRNA, they are also the easiest to understand. Okay, a key feature of these splicing mechanisms, particularly the group 1, group 2, and spliceosomal, is this transesterification process. In this case, you can see what we're doing here is we are essentially doing a nucleophilic attack on the phosphate group with a phosphodiester bond and breaking a phosphodiester bond. We can look at this 2 ways, but essentially you can see a very good leaving group here during this process is one of the nucleotides. A new nucleotide is making a bond isoenergetic thermodynamically, so you can see that no energy has to be actually be invested here. From an organic chemistry perspective, which is really what's important, you can see that the nucleotide is coming in as the nucleophile, a nucleotide is leaving, in a sense, as the leaving group, right? So this is what we call the transesterification, essentially replacing one phosphodiester bond with another. This type of chemistry will be really pivotal in terms of understanding how these group 1, group 2, and yes, also spliceosomal introns actually work. Okay, so here is our group 1 intron, showing you 2 different representations of it. A simple one here and a slightly more complex one, but in both cases, still representing the nucleotides is just letters. I would encourage you when you're kind of practicing this and learning this at home, I don't think you need to write out hundreds and hundreds of bases. I do think you should probably try to see the chemistry from a structural perspective by yourself. I think that would be worthwhile. Otherwise, it's very easy to remember lines and pictures and memorize images. More difficult to answer questions about those things if you don't really understand what's happening. So even though I'm showing you some nice condensed images here, the basic idea that I think you should do is actually go home and start practicing with some structures, because that's much more likely to give you the type of deep insight that will allow you to answer questions about these on the exam. That's my little fun. Here we go. What do you need to have a group 2 intron? Well, in the simplest case, you need to have 2 exons, right? An intron between them that has to be spliced out, but you also need something else, which is that you need a G nucleotide. Now, just to be clear, this is something that every year a couple people mistake, so let me try to get it out of the way here. Not GTP. This is not GTP here, this G, okay? This is an isoenergetic self-splicing reaction. No external source of energy is required, okay? So that is not GTP, that's just G, right? That's just the nucleotide base with its sugar, okay? So no GTP is required here. It does, however, have a nucleophile. That nucleophile, the 3' hydroxyl group, will do an attack on the phosphate group on the 5' intron, and you can see that it will essentially liberate the first half of the intron. Right? The first half of the intron is now liberated with the G attached. In the second step of the chemical reaction, this is true of all group 1 introns, in the second step of that chemical reaction, the free hydroxyl group from the 5' intron will attack the phosphate group that starts the 3' intron, and that completes the splicing. So the leaving group there is that entire intron, which has gotten one residue longer because of the use of this external nucleophile.
What is the G?
G, yes, well, but we just write it as G monophosphate, yes. No, actually just the just the other phosphate, so it's a nucleoside. Okay, good question. Here you can see again, represented here with the external nucleophile doing the attack. There's the intron there in the first step, liberating the 5' end of the intron. And then in the second step, the nucleophile, there's the hydroxyl group at the end of the first exon, attacking the phosphate group there at the start of the second exon. This is what we call group 1 splicing. It's a common mechanism. There are sequence variations within the actual introns themselves, but what there is not any variation at all is the requirement to having this external G. Okay, so that is the key pivotal thing, is you must have an external G. Now, before I go forward and show you the group 2 intron, this might look like, okay, well, all I need to have is some DNA and some sort of an intron between there and put in a G, and this is going to happen. But you'll remember from organic chemistry and even our discussions in biochemistry, the importance of aligning nucleophiles and electrophiles in the right direction to have these nucleophilic displacement reactions happen is really quite critical. So even though this is kind of how we draw it out, so you see the chemistry, I'd like to show you the actual structure of the shape of the intron so you can see how complex it is for the actual RNA to fold into to have that happen. And so this is what you're seeing here now, right? This is the intron with little pieces of the exons there also. Here's the secondary structure diagram with multiple stem loop structures. Here's the three-dimensional structure. The critical G here is shown in pink. This is what the RNA has to fold into for this reaction to happen, just to position the G in the right position so that it can do that first nucleophilic attack. So even though that image may represent or look very much like, oh, you just have a G and it comes in and anything can splice, you have to have an RNA that actually has a sequence that can fold into this particular shape for the splicing to actually take place. So that is the group 1 intron. To show you the group 2 intron, I actually need to introduce a sailing term. We don't do much sailing in this class, but every so often, or for those of you that are interested in knots. This is a lariat. So a lariat is a three-way structure and a knot. You probably generated one of these when you tied your shoes, so it's not that exotic of a creature. But I'm going to keep referring to the lariat, the lariat, the lariat. This is the basic shape of a lariat. Okay, so it's this three-way structure that you see here. Okay, so why do we mention the lariat? We mention the lariat because the other name for the group 2 intron is actually the lariat maker, because as you're going to see, the group 2 intron is going to make lariats. The first thing that you'll notice is, again, no external ATP or GTP or anything. You're right, by the way. Mr. Corsair, correct? But none of that here at all. There is no external nucleophile. So when you have a group 2 intron, no external nucleophile whatsoever. So not only is there no GTP or ATP, there is no GMP doing any of the chemistry actually here. So that means we need a nucleophile to get the whole process started. What is that nucleophile? Well, in the case of group 2 introns, that nucleophile is going to be an internal base that is present within the intron called the branch point adenine. Okay, so you can see here, we have the branch point adenine. That is going to be the nucleophile. That is a very problematic nucleophile. And the reason why that's a very problematic nucleophile is, remember back in the group 1 intron where we had the 3' hydroxyl group free that made a very good nucleophile. Here we don't have a free 3' hydroxyl group because that's all tied up. So when you're seeing that little hydroxyl group descending or coming off of that A, that is not the 3' hydroxyl group, leaving the only remaining nucleophile that's actually present, the 2' nucleophile. So the result of this is when you do that same nucleophilic attack to free the 5' end of the intron, you end up with that lollipop, or I've heard it referred to as a balloon-like, but let's just call it by its scientific name structure, a lariat. So you see here how we've actually generated a lariat. Why have we generated a lariat? Because for that A, again known as the branch point A, both the 2' hydroxyl and the 3' hydroxyl are locked up in phosphoester bonds. So this is why you can see a little more complex image there, where the actual nucleotides end up during the splicing process. It's okay for now just to focus on here. In the end, of course, there has to be an actual splicing taking place to free the entire intron. That happens when the 5' hydroxyl again attacks the 3' phosphate. So the second step of this is actually the same. Right? There is no distinction, in other words, in how the second step of the chemistry happens. The first step is due to the absence of an external nucleophile. So you have this 2' hydroxyl that is properly positioned. And just as though it is always a G with group 1 introns, it is always an A with group 2 introns. Okay, so just to be clear, it's an A. Again, just to show you again, that isn't just going to happen. It's not a matter of just having sequences, but also having proper structural arrangements here. Here is what the secondary structure of the group 2 intron is. That is the shape that I'm showing you here that is required for this to actually fold into to properly position that branch point adenine with its 2' hydroxyl, that it can do the first attack on that first phosphate group and open up the start response and the splicing process. Okay, so there are individual domains here. Each one of them is particularly important, but the overall take-home message from this is it's not just a bunch of nucleotides in a row that are capable of doing this. You really do need all of those sequences to form these particular secondary and yes, tertiary structures in the RNA for this type of splicing to happen. And if you make mutations such that these structures are not present, even if you have the branch point adenine and all these other residues, this is not going to happen. And that brings us to a very, very interesting point. Because where we see group 1 and group 2 introns are in general relatively simple things like protists, for example, where there's some relatively simple organisms. We have a lot more introns than they do. And our introns have had a lot more difficult time, opportunities I should say, to evolve just by natural sequencing variations, as well as the fact that there are in fact useful things that are found in those introns. And this creates a bit of a problem, right? So if you just have very highly conserved introns, having the sequences to fold into this shape is not such a big deal, right? Even if you have a mutation here, you could perhaps have a compensatory mutation, and splicing can still take place. But we have so many more proteins, and so many more introns in those proteins, and so much greater chemical complexity to those introns, that group 2 splicing should be impossible for us. Not because the chemistry won't work, right? But rather because keeping those sequences fixed like this over the course of evolution is quite stringent. And it would also mean that our introns never could be used to do anything, which, as we'll see later, sometimes useful things do come out of introns. Not protein things, but other things. Anyway, this is a problem, and yet we somehow managed to splice. And the answer is, well, how do you possibly do this type of splicing mechanism without having this high level of sequence conservation throughout the whole structure? How, in other words, do you have an organism like us that has whole chunks of its genome that just haven't evolved? And the answer is to have something called the spliceosome. So here is the features of the spliceosome. The spliceosome has many, many, many proteins. And notice I said many, many, because there are well over hundreds and perhaps more proteins that are actually being required. Despite the fact that I'm only going to show you 5, those are the core proteins that are required, but there are many other proteins. And an interesting feature of this enzyme called the spliceosome is that the protein composition isn't even going to stay the same during the entire splicing process. Some proteins will come in, help some of the process actually happening. They'll leave, other proteins will come in and replace it. So this is a very, very sophisticated machine. In addition, the spliceosome will include ribonucleoproteins, so some of those proteins will actually have RNAs bound to them other than just the ones that are being spliced. Finally, you're going to see some ATP energy get used here. Some use GTP, but it's the same point, nucleotide triphosphate. So with all of those details in mind, the fact that group 1 and especially group 2 did not use proteins, did not use any energy, had highly conserved sequences, the most important takeaway of all is the splicing chemistry is exactly the same. There's no difference in terms of the mechanism by which the splicing happens with the group 2 intron and the way your introns get spliced. Chemically wise, they're exactly the same. So why is it that we need to have nucleotide triphosphate energy, and why is it that we need to have this enormous array, including the 5 proteins I showed you here, but vastly more of them? And the answer is, your introns aren't super well conserved. So the purpose of the spliceosome, strictly speaking, is not so much to splice the RNA, the purpose of the spliceosome is to drive the RNA using certain conserved sequences within positions into the conformations that are required for splicing to happen. And that is what the energy of the spliceosome is actually being used for. The energy is not actually driving the splicing itself. The splicing is still an RNA-dependent process. The spliceosome is really there to drive the conformations and the conformational transitions that are required for each of those discrete steps, to drag the branch point adenine down to where it can act like a nucleophile, to hold on to the entire complex, to assist the first part of the lariat from being removed, to drive the second conformation to put the nucleophile on the first exon into the right position on the second exon phosphate where it can do the attack. And so that's really what's significant. Just in terms of chemistry, everything is exactly the same. The key is that there are certain conserved sequences within the spliceosomal introns that get recognized by the spliceosome in different steps. So those, of course, have to be particularly well fixed. And the spliceosome is going to do essentially all the driving things into the right conformation. So again, there are a number of proteins that are required. I'm not asking you to memorize the names of any proteins, but I just wanted to show you that there are a substantial amount of nucleic acids. So these are actually RNA binding proteins that you'll actually see. The RNA is in fact doing a lot of the aligning. The most conserved sequences that you will see will actually be between RNAs that are in the spliceosome. That's the U1 RNA. You can see the U2 RNA there. So those are RNAs, but they're not free RNAs. They're RNAs that are part of protein complexes. And those are what's going to essentially base pair up with the right positions on the RNA that is going to be spliced, the intron, and bring those positions together. So the recognition of the RNA to be spliced is by RNAs that are part of the spliceosome. So it's really RNA doing all the hard work here. The proteins are just there to support it. So we see here two important features that I want to highlight. You're not, again, being asked to memorize any structures here, nor really the names of these RNAs. So that's not what I'm asking you to zoom in on or why I have this slide. I'm asking you to have this slide because, again, you can see, first of all, there are these very highly conserved sequences. These cannot vary. These are required to say, hey, this is an intron, splice it, right? You can see at the 5' end, one of the really interesting features is that, well, okay, I've seen these nucleotides before, right? I mean, I know what a U is, a C is, a C is, an N. Okay, what is that? Told you that a lot of these RNAs are modified RNAs. This is both important, obviously, functionally, but makes it difficult to study. Here's your first modified RNA that's actually had some function. This is an RNA base called pseudouridine. Have to absolutely have pseudouridine in the U1 RNA protein complex for this to happen, and that's going to align with the 5' end. Here you can see all the way to the 3' end of the exon. Very close to it is that branch point adenine that's kind of been pushed or forced out by the protein complex. The second level of recognition that's actually as well, these conserved sequences at the end. These are what these proteins are actually binding to, and now the protein complex is going to come in and drive this into a conformation where that A can catalyze that attack on that G, right? So this is what's happening. So again, I'm not asking you to memorize the names of any of these spliceosomal proteins, just to be clear. They are here because, otherwise it'd be even more confusing. But I'm just showing you again, schematically, ATP is being used, right? That ATP is not for the splicing reaction. We're not making, phosphates and phosphate leaving groups. That ATP is being used to drive conformational changes in the spliceosome, allowing proteins to bind and the RNAs to fold into their particular conformations. We can see through the process how proteins are coming in, driving those sequences that are far apart in sequence but are now close in space to properly position the hydroxyl group, the 2' hydroxyl group on the A, close to the end of the exon. At some point, we have an active spliceosome, so the chemistry can actually happen. This requires the loss of some protein components. And in the final step, after we do the second nucleophilic attack, which of course is requiring another set of conformational changes, some of those proteins are actually helping remove the intron, essentially taking it out of the spliceosome, so the intact mRNA can be made, and RNA can be made. Okay? So again, don't worry about the names, worry about the ideas here, worry about who's recognizing, like these RNAs. The recognition, presumably, is not very much of the protein, but by highly conserved RNAs that are found within the spliceosome, and the energy is being used essentially to drive conformational changes not make leaving groups so that you could reform phosphomaster bonds. All of that is RNA-dependent chemistry. Okay, so far, so we can kind of do a comparison. Here's our comparison table. The only one that strictly speaking requires protein, of course, is the nuclear splicing. There are some group 2 introns that have been discovered in association with proteins, but in that case, that seems to be much more of an early evolutionary event where it's helping stabilize the structure. There's still no examples of where the proteins are playing any direct role in the splicing process. So really, for the most part, nuclear and group 2 splicing are the same. It's just there's no possible way that you can constrain evolution in an intron, our introns, anyway, to keep the structures. So the splices open us to grow apart. Here, I'm showing you again, just as an example, we started with a primary transcript. We actually started with the DNA, we made the primary transcript, capped it at the tail at the end. We removed a lot of the introns, and so you can see the relative comparison in the typical size, starting with about 7,700, all the way down to about just 1,800 nucleotides, which will actually get used. This, of course, is excluding those 250 or so bases that are present just in the A. The actual size of the coding gene is obviously shrunk down quite a bit. It is possible to make multiple different genes even with the same coded mRNA, and this is due to processes that are known collectively as alternative splicing and poly-A choice. So these are related, and in fact, they can be easily confused because they often come in combination with each other, but let's treat them a little bit like they're somewhat different. I think the easier one to understand is the case of alternative splicing. A typical gene will contain a handful, many, 5, 10, or even more, exons with introns between them. Now, in some cases, all of those exons are spliced together to make one gene, right? So if you have 5 exons, you splice exon 1, 2, 3, 4, and 5, pop out all those introns, and you have the largest version of that mRNA, and then you can go and, like things we'll talk about next time, make protein out of it. But sometimes, not all of the introns are actually used. Sometimes we have something, a case where we have exon 1, we don't use exon 2, so we connect it directly to exon 3, and then exon 4 and 5 and 6 and so on. Right? So you can make multiple different coding sequences out of the same RNA by not including or including certain exons. Right? So the splicing process does not always remove all the introns. Sometimes it takes out an intron with an exon as well. And that's a viable possibility. And it allows you to code for different proteins using the same mRNA sequence. The key, and a really important key here, is that despite the fact that you can include or exclude certain exons, they always have to go in order. So you could have 1, 2, 3, 4, 5, 1, 3, 4, 5, 1, 3, but you can never have an RNA, an mRNA, that's made like 3, 1, 2, right? Or 4, 2, 3. They always have to go in the order in which they are found. Some can be excluded, none can be excluded, but they always have to go in that order, and this allows you to diversify the number of proteins again that you can make. There is a related phenomenon known as poly-A choice. Also in poly-A choice, you have a situation where you can make multiple versions of a protein using the same basic mRNA sequence. But in this case, it's not due to splicing. In this case, it is due to the presence of multiple poly-A sequences within the same coded gene. So I showed you when we looked at poly-A, you had that AU-ish sequence and that GU-ish sequence, right? Normally, that would appear only once, all the way at the end of the mRNA. But it can turn out that there may be multiple copies of that sequence that are distributed, and then additional introns and exons, and then another poly-A sequence. And so what you can have is shorter, for example, and longer versions of the same coding sequence. So we can see a primary transcript here, which just for simplicity, we haven't shown any introns or exons, but these are not exclusive. You can see that there are 2 different possible ways and places in which the poly-A tail can be made. Either of those, in theory, can be chosen. If you choose the A2, you you make the longer version of this protein. If you choose A1, we make the shorter version of this protein. Now, back in biochemistry 1, one of the things we talked about was the idea that to start the process of all of these antibodies being made, an antigen would have to bind to an antibody that was attached to a B cell by a transmembrane domain. And then later, the B cell, upon binding to that specific antigen, would release soluble versions of that protein that were essentially identical, except they were missing the transmembrane protein. So we have two proteins, almost identical, except one has a transmembrane protein and the other does not have a transmembrane protein. It would be very, very inefficient for nature to put both of those proteins into the genome, the one with the transmembrane domain and the one without the transmembrane domain. So that's not what nature did. Nature took that gene, and yes, there are introns and all these other things that we talked about, but let's just focus here on this one, and essentially put in two poly-A sites. Initially, to make the transmembrane version of the protein, it uses the second site. All of the information between A1 and A2 codes for the sequence to make a transmembrane protein. But then, when it wants to make soluble versions of antibodies to release out into your circulation, into your plasma, so that you can fight infections, it switches to using the first poly-A site, in which case it ends after A1, that's where the poly-A tail gets added, in which case there's no transmembrane domain present, so you have a soluble protein. So you can see here that by both poly-A choice and by splicing, one could make a whole different varieties of proteins, in some cases having very, very different properties. The extreme version is probably the one I just told you about, having a soluble versus a membrane protein just by using these options that are present. Okay, we will get back to the process of splicing when we look at tRNAs a little bit more, but I want to show you one additional major way in which the sequence that is initially coded for the DNA might be different than the information that ends up being coded in the mRNA that makes protein, and this is a process that is known as RNA editing. It also, by the way, answers the question that we addressed all the way at the beginning of this lecture. If octopuses are so awesome, why are they so awesome, and why are octopuses not running the entire world? So those are the questions we're going to answer. What is RNA editing? How does it help us? And most importantly, what does it say about octopuses? Because I'm very interested in that. Anyway, here we go. RNA editing is the process of making specific chemical changes to the mRNA that result in it having a different sequence than the one that was originally coded for by the DNA. So these are enzymatic processes, and they essentially come in two different flavors. One of these requires a type of RNA to be present called a guide RNA, and that guide RNA is going to serve to take an enzyme called an editosome, position it in the right position in the mRNA, and make specific changes there. So the editosome is going to contain another RNA, not the one that you're modifying, but another RNA that is coded for in the genome. It's going to bind to the mRNA that you just made, and it's going to use that enzyme, it's going to use the information in the gRNA to fix the mRNA to have a different sequence. Generally those sequences will involve either inserting nucleotides or deleting nucleotides from the mRNA, which of course will change the coded information within. There is a second type of editing, and this is called base substitutions. In this case, the mechanism of recognition is still actually something that's under investigation, but it does not require the presence of a guide RNA. Conversely, the types of changes that are made are very, very limited to the types of chemical changes to bases that, for the most part, we already saw when we looked at DNA damage. So you can imagine that what these substitution base edit changes are doing is actually intentionally damaging the sequence of the mRNA by making chemical modifications to the bases, which in turn changes the information content within. So here are, I should say, just representative examples. You can see, I know we have not done the genetic code yet, but hopefully you guys have seen a little bit of this in bio, so it will make some sense. Up here, we have a DNA that codes for this specific sequence, right? That is the DNA coding strand. That's the information that would actually, the sequence that would result, I should say, from translating that strand through the RNA process. By bringing in a guide RNA, such as the one that you actually see here, so there's a guide RNA that can both result in insertions and deletions right, to nucleotides that are present by the enzyme that is there, you actually end up with the edited mRNA, and you can see we've actually made sequence changes. The mRNA has been structurally changed by having nucleotides either added or deleted with the instructions, I should say, of that guide. So it's not arbitrary. This is not a random process. And again, what this is allowing your genome to do is to function a little bit more efficiently. You can have one sequence for making one type of protein under certain circumstances, and if other circumstances were to arise where you need another protein, instead of completely recoding for it, you take that initial bit of genetic information, turn it into an RNA, bring in your editing factory, and make a whole new RNA sequence. So this is essentially the first type. The second type, not surprisingly, because we focused on it so much in this class, is if you want to change the sequence, the easiest way to do so of a single base is just to deaminate it, right? So this is a reaction that can happen anyway in water. It's fairly frequent, but there are enzymes that will actually come in and cause deamination. There are adenosine deaminases and cytosine deaminases. These will also change the coding sequence of the mRNA. In our case, what these are quite often actually used for is part of the antiviral response. If you have a viral RNA that gets into you, one really good way that your body has to defeat the virus is to intentionally change its sequence by deaminating it. But these are also used to produce functional mRNAs as well. So they're both antiviral, but also used for making essentially one sequence behave a different way. Anyway, let's look at the first one. Again, this is using that first strategy, using an editosome. We can see here again the DNA sequence, that would be the coding for it. We have the pre-edited mRNA that ends up in. We can then bring in a guide RNA. The guide RNA in this case can result in the addition of U's. In some cases, it can result in the deletion of U's. And so as we said before, we have this one sequence that would be coded for by this DNA into mRNA in the absence of the editing. And if we bring in the actual editor, it pairs the sequences up. In this case, you can see it's actually inserting a couple of additional U residues, and that changes the actual protein sequence that it produces. The best study of this is actually nothing to do with the Egyptian stuff. It just seemed kind of cool at the time. It's called the tuttase. You can see the way the tuttase actually works. When the guide RNA is paired with the pre-mRNA, there can either be bulges that happen because there's more nucleotides in the pre-RNA, and those we can actually recognize, or in some cases, the guide RNA will be slightly longer, in which case that's a signal to do insertions. So the tuttase is capable of actually cutting the sequences in those positions, either excising extra bases that have not paired with the guide, or if the guide is a little bit longer, cutting the DNA and inserting those extra nucleases and sealing it back up. At that point, another enzyme, which is an RNA ligase, can come in. All of these cuts and deletions and insertions that can be made require sealing. That RNA ligase is essentially working using the same chemical mechanism as the DNA ligase we saw. So this is the function of the tuttase. Again, the other possibility is not to make specific to encoded changes that are present in a guide, but a different class of enzymes. These are RNA editing by base substitutions. There are two classes of these base substitutors. One of them work on adenosine and deaminated and inosine. Inosine gets read, in generally speaking, as a G, so we get an A to G tag change, which is obviously quite dramatic. Again, this is in the RNA now, not in the DNA, so we're not changing the genetic information. There are also cytosine deaminators. We actually saw this when we looked at the immune system, when we talked about B cells a little bit. But here we have a C that's going to be coded for by a U. So these are another way to take the same sequence, right, and change it after it's already been transcribed into RNA. That sequence, in this case, may result in changes of a single amino acid or two, but that can actually be quite dramatic in terms of the functional properties of the protein. Anyway, this brings me to what I really want to talk about, you can probably tell, which is octopuses. Because what does this have to do with octopuses? It turns out that we definitely have base deaminators. I mean, I just want to be clear about that. We do so, although most of the time when that happens in our genome, it doesn't actually occur within coding sequences, but rather it occurs in non-coding regulatory sequences. And so these might result in the protein being made more or less. It's really hard to predict. But we do this. But we don't do it by very much. And the reason why we don't have to do it by very much is that we have so many other options for diversifying our proteins. Really not necessary for us. Who edits their genomes like crazy, their RNAs, are octopuses, and this is using the second approach. And one of the amazing things about octopuses is how dramatically and quickly they can adapt to amazing differences in their environment. They can change rapidly in response to temperature, the pressure by which they're occupying in the ocean. They change essentially the RNAs constantly. So they very heavily edit their genomes, which is a good thing, because it means an octopus can go a couple hundred or even more than that feet in water and pressure difference and very quickly make proteins to help them deal with that. They can change their temperature, which they are in the ocean dramatically, far more than we could ever adapt to. And so that answers the question, if octopuses are so good at doing this, and editing your RNA is such a powerful tool, why don't we do this more? Or more importantly, why aren't octopuses in charge? And the answer comes back to the fact, again, that it's not any sequence that can get edited, but the sequences around the base that get edited have to be highly conserved. These deaminators are not just randomly running around doing this. There are certain conserved sequences where those deamination events are going to take place, not just in the edited base, but around it. What does that mean? It means that if you don't really rely very much on editing, your genome can evolve, right? Who cares what's next to this A if it's better for you to change the base next to it during the course of evolution? If it's adaptive, you do that. But octopuses basically cannot evolve because so many of their adaptations depend on editing, and because not only the edited base must stay intact, but the sequences around it. Two very different species of octopus have much more closely related DNA than two species of other types of organisms. Editing made them fantastic at adapting because they could rapidly change the functions of their proteins. But it also creates an evolutionary dead end because you're stuck with that sequence. You cannot allow that sequence to evolve. You cannot allow that sequence to have insertions or deletions or mutations. So editing is great, but it creates evolutionary dead ends. Again, because not only the edited sequence you're depending on staying the same, right, but all the sequences around it. So octopuses are kind of stuck in this evolutionary trap of really not being able to allow their genomes, the sequences in their DNA, to change by very much. Okay. So where are we with us? Right, we were talking about these RNAs. These mRNAs are going to be, of course, made and spliced up in the nucleus, but we're going to do everything else. It's going to take place, or a lot else anyway, is going to take place in the cytosol, which means that both things have to be able to get into the nucleus. That includes the raw material, but also the proteins. And that also means that some things have to be able to get out of the nucleus. So we talked a little bit about previously this idea of the nuclear pore complex. So just remind you again, this is a large complex of protein in which there are thousands of them within the nucleus itself. They look like little holes in the nucleus, but they're actually protein complex. Here's actually a microscopy image. So these things are large enough that they can actually be visualized by light microscopy. So these are not small structures. Very high resolution light microscopy, but light microscopy is all the same. And you can see they're basically scattered around the nucleus, and this is where anything that is going to enter or leave the nucleus is going to go through. Some things kind of get in without any problems. Small things like a nucleotide or even a really teeny protein in some cases can sneak through the nuclear pore complex. But for the most part, the process of getting things into the nucleus or out of the nucleus is very heavily regulated. The protein has to bind to a component, which you want to say bring into the nucleus, bind to the nuclear pore complex. It has to open up a protein gate and allow that to be emitted. So, if you want to send a protein into the nucleus, how do you do so? How does the cell know, yeah, this protein belongs in the nucleus because we're going to need it for splicing or making DNA or something? No, not that protein. Keep that one out of the nucleus. Well, it turns out that at the C terminus of proteins that are made in the cytosol that are destined to enter into the nucleus will have what's known as a nuclear localization signal. It's not absolutely conserved, so it doesn't have to be this. It's just generally a run of 4, 5, or more positive charges at the end of your protein. When you have this, at the C terminus of your protein, it's going into the nucleus. So So a dense positive charges of arginines and lysines, with occasionally being interrupted by hydrophobic residue, is going into the nucleus. Which means that if using genetic engineering, you attach this to pretty much any protein that's not supposed to go to the nucleus, it's going to the nucleus. And so this is called the nuclear localization signal. There is also, we'll see, a nuclear escape signal for proteins as well. So yes, so we want to talk a little bit now, before we go further, into this process of transportation, getting things necessarily to the right place. The proteins that are involved in this transport are collectively known as the karyoferins, and not surprisingly, they can be divided into two families that work roughly similar to one another, but obviously do exactly the opposite thing. We have the exportins and the importins. So exportins will bind to nuclear RNAs, which are almost always associated with other proteins, and send them out. Again, really clear, the RNAs aren't going out by themselves. So we'll see that the exportins have to export nuclear RNA complexes, even if those RNAs are meant to function in isolation. And similarly, we have importins. These importins carry cargo RNA, again, typically bound to proteins, into the nucleus through the nuclear pore complex. So if you have an RNA and it's in the cytosol, say, you want to get it into the nuclear pore complex, you need to have a protein, first of all, that it binds to, and that protein should have a nuclear localization signal. Okay. Again, yes, so these are the signals: the nuclear export signal, that means go out; nuclear import signal, that's bring the sequence back in; and the nuclear import signals are much, much more highly conserved. Another thing that we're going to need to do, therefore, is to have a protein to function as a switch, right? What do we mean by a switch? Well, we're going to have an RNA bound to a protein, presumably tightly enough that they're going to stay associated. It's going to be in the nucleus, but we want to get that RNA out of the nucleus. Then we need it to be able to actually dissociate or to fall apart. And so this is going to require us to have some sort of a switching mechanism that will say, hold on to me under one set of circumstances, release me under another. And this here that I'm showing you now is a switching mechanism that is used not only in this specific process, it's actually a pretty general switching mechanism that we'll see before, and it's based upon a class of proteins that are known as G proteins. So what are G proteins, and how are they functioning as switches, and then what are they possibly going to have to do with anything regarding getting RNA protein complexes out of the nucleus? So the one I'm showing you here is RAM, but this is actually quite general. The key to these complexes is the following. First of all, RAM will have different conformations, whether it is bound to GDP or GTP. In this case, this RAN protein in the GTP bound form will have a certain affinity for the RNA and a different affinity in the GDP bound form. So by binding to GDP or GTP, the shape of this protein is, oh, fine. That's really so rude. 15 points in there. Sorry about that, guys. Okay. So we have protein that can have two different conformations based on whether it is bound to the GDP or to the GTP, and that conformation is significant enough to affect its affinity for other things. So there's our switch. But what is GDP or GTP having anything to do with it? Well, it turns out that in addition to whatever functions we'll see it have, RAN is also a GTPase. So if you bind a GTP to RAN, it's going to hydrolyze it to GDP. So RAN has now at least two clear functional differences. One, to bind to something, differently whether it's bound to GDP or GTP, and two, hydrolyze the GTP to GDP, so it's an enzyme. But what type of enzyme is it? It is a really pathetically bad enzyme in two respects. First of all, the first respect is this process of hydrolysis is going to be really slow. So you remember we talked about like turnover numbers back in Biochem 1, and we saw like 10 per second, 100 per minute, 1,000 per minute. This has a turnover number of exactly 1. 1 GTP will get converted to a GDP. So that's one of the things that makes RAN a really, really, really bad enzyme. It's really slow and bad at doing its job. So clearly its purpose is not making GDP. What's the other thing that makes G proteins like RAN such bad enzymes, although, as we'll see, very well suited to what we need it to do? It is product inhibiting. So that GDP is the product of the RAN hydrolyzing the GTP to the GDP. It's not coming off. It's stuck. And of course, if the product is stuck on the enzyme still can't do another round of catalysis. So G proteins act as switches by having two different conformations in which they can bind to other proteins, depending on whether they are bound to GTP or GDP. But significantly, they can hydrolyze the GTP to GDP slowly, and when the GDP is present, it can't come off. So this means that in addition, we do need some other proteins as well. For example, this process of hydrolysis is slow, but it can be sped up by bringing in another type of enzyme, in this case a non-classical enzyme called a GAP. A GAP protein can bind to G proteins like RAN and squeeze them. I kid you not. Sounds like an analogy. It squeezes them until the GDP falls off, so it can be replaced by the GAP protein. Let's wipe that up. The GAP protein, the one on the top, was I was making the point to, squeezes the GDP off so that a new GTP can bind. Okay, so we have a GEP enzyme, which is a non-classical enzyme. I apologize for thinking ahead. The GEP enzyme knocks the GDP off so that a new GTP can bind. In addition, to speed up the reaction, to have it happen a little bit faster, that's where we have a GAP enzyme that helps the RAN by coming in and assisting it in facilitating its hydrolysis to bring it into the GDP state. So this is what we call G proteins. We have a G protein itself, which again, two different conformations, one of which can be associated with certain proteins, the other which it cannot, and that is determined by whether or not it has GTP bound to it or GDP bound to it. It is a product inhibited enzyme. So itself, not only is it slow, but it cannot lose its product. The only way to lose its product is by having another enzyme, a non-classical enzyme called the GEP enzyme, come in and displace the GDP so another GTP can bind. And we need to have a GAP enzyme. The GAP enzyme actually speeds up the hydrolysis process, which otherwise would be way too slow if it was left just to random. But anyway, we seem to have diverted very far from RNA. So let's try to see how we actually get things out of the nucleus and what that's actually going to require. The first thing that's going to happen, of course, is we made our RNA, our cargo RNA, it's going to have to bind to a protein. We have no way of getting the RNA directly out of the nucleus directly, right? So it is going to have to bind. In addition, that cargo protein is going to have to bind to RAN. If that cargo protein is bound to RAN, and the exportin is bound to RAN, then now that is the signal you could leave the nucleus. So what do we need to have? We need to have RAN, which is bound to a GTP, the exportin, and the cargo protein that collectively have our RNA associated with it. That will bind to the nuclear pore complex, which will open up and allow the whole thing to leave. Once they leave, of course, we don't want this whole complex to remain associated. We want to release our protein bound to the RNA. We need to do that by causing the GTP to be hydrolyzed to the GDP. That causes the entire complex to go from a high affinity state to a low affinity state, and that's going to now release our protein RNA complex. Okay? So this is what we call the RAN cycle. Getting everything back into the nucleus, of course, is going to take importance. We'll talk about that after we talk about protein translation. So this is the key feature, right? We start here with RAN in the GDP bound state. We need to swap that off, right, using the GEP protein, replace it with GTP. At this point, we bind to our exportin, we bind to our protein cargo, RNA cargo complex. That is the signal to the nuclear pore complex. You can leave now, opens it up, allows it to leave. We need everything to come apart once we are actually in the cytosol. In the cytosol, right, we have our GAP protein, speeds up the hydrolysis of the GTP down to GDP. That causes the affinity of these proteins to become looser for one another, releases everything, and now we have our RNA protein complex present inside of the cytosol. This is the generic mechanism by which these things actually leave. To be clear, after all of this, and this is the part, RNA, mRNA, actually leave by a slightly different complex of proteins, which really functions by very similar mechanisms, so there's no necessity to go through everything, but it's actually called the T-Rex complex. So the fundamental mechanisms by which this work, including the going through the nuclear pore complex, are largely the same. But just to be clear, the actual complex of proteins that are responsible for bringing mRNA that you made out of the nucleus so that something can happen to them, that's called Tregs. And Tregs has a really, really important feature to it, which is that it will not allow unspliced proteins to actually exit the nucleus, under the notion that if it's not been properly spliced, then probably there's something wrong with that RNA. So how does the Tregs protein complex know that the actual splicing has happened? It just looks chemically like an RNA. During that splicing process, when the spliceosome is actually working, after the introns are actually excised and the exons are spliced together, portions of this proteins are actually left behind on the exon-exon junction. So if you, after you splice, rather than take everything off, proteins are actually left at the splice sites, and those proteins are recognized by the Tregs complex as a signal, yeah, you can actually leave the nucleus, because you are a spliced RNA. And so that's really important. Otherwise, those unspliced proteins would not be able to exit. Similarly, the cap has to be in place, and the poly-A has to be in place. If that's not the case, again, not recognized by this Tregs complex, and you're not leaving the nucleus. One thing that is both really cool and I guess really awful at the same time is how HIV mRNAs actually get to leave the nucleus. Because back when we talked about how small space there was, there's nothing smaller really than being inside of a virus. And if you're a virus, particularly one as complex as the HIV virus is, you've got to pack as much possible information as you can into your small genome. And the way some viruses like HIV do this is actually to have coding sequences within their introns. So functionally speaking, for such a virus, there are two sets of proteins, right, that can be encoded. The spliced ones and the unspliced ones. But this creates a problem, of course, because if you're not spliced, you shouldn't be able to bind to the Tregs complex, you shouldn't be able to leave the nucleus. So how does HIV get around this? How does HIV manage to send out both spliced and unspliced mRNAs? The answer is this protein here that's present in the HIV, which is the Rev protein. It actually binds to the introns and the receptor in the nucleophore complex and fools it into thinking it's essentially a splice junction protein, right? So one of these exon junction complex proteins. So the HIV protein, this Rev protein, binds to it and mimics effectively having a splice junction protein there, and that in turn allows it to export both spliced and unspliced mRNAs, which effectively doubles the amount of information that's capable of being stored in such a small genome. Anyway, a couple of things, just one or two slides about these to keep it relatively short. In many cells, particularly larger cells like neurons, there could be a pretty tremendous distance between the nucleus, where these mRNAs are first being made, and the part of the cell where you actually want to use that mRNA and then eventually translate it into proteins, allowing it to actually move by passive diffusion to get from one position to another position would be pretty ineffective, just eventually hoping that the RNA actually gets to the right place. So there are also RNA transport proteins that are actually capable of functioning like motor proteins, binding this RNA cargo and actually delivering it to places far away where the ribosomes, which we'll talk about next, actually are, and doing the translation there. So this is something called local translation. Again, these are proteins that are capable of binding the mRNA, and particularly in cells like neurons, which can be very long distances, actually bringing the RNA to those positions where they are translated where they are needed, so that they're not requiring this passive diffusion to actually get them there. Anyway, look, all good things have to come to an end, and bad things too, of course, but at some point the RNA is just not going to be needed anymore, particularly the mRNA, and it needs to be destroyed. Some RNAs, we pointed out, particularly in prokaryotes, might have minutes or even less than that lifetime. Others can actually persist for a couple days. That, in large part, will determine on sequences that are actually present in the RNA. We'll look at this a little bit later. But these sequences at the 5' and 3' ends can have a very, very substantial impact. There are complexes, both in prokaryotes and in eukaryotes, of nucleases that will actually begin the process of destroying an mRNA if it's not needed. First by cutting it up into smaller pieces, and then nucleases coming in and actually degrading those. In you, there's actually a larger complex set. This actually starts with the one enzyme that's capable of hydrolyzing that odd 5 to 5' bond. That's called the decapping complex. There are also specialized complexes that will work from the 3' end, capable of degrading the tail. mRNA has its fate to come to this large complexes of nucleases known as the exosome. It's just like a chopping machine for mRNAs, right? So there are multiple nucleases with different specificities there, and they just grind through the mRNA and essentially degrade it up into pieces. So that's where your final activity of your mRNAs actually will be in the exosome, a machine that essentially degrades us back to nucleotides, which presumably can be used. Everything so far has been about mRNAs, but we should also take a few moments to look at the other major class of RNAs that we're going to need to make proteins. And this is tRNAs. And much like mRNAs require a substantial amount of processing, tRNAs are also not made in a form that's chemically viable or ready for them to be used. And so they will require some fairly exhaustive processing as well, some of which just actually seems quite illogical. Initially, for example, the primary transcript of a tRNA that's been read off, and this is these called 3's, have segments at 5' and 3' ends that have to be removed. And there are specific enzymes that will recognize the 5' and the 3' ends of these tRNAs and chop them off, right? So we can see RNase P working here on the 5' end, RNase D working on the 3' end. The one at the 5' end is slightly more interesting because it's actually a ribozyme that's cutting a tRNA. It will process them off. We also will have introns that need to be removed, but by a slightly different mechanism. Base modification enzymes will actually come in and take some of these bases and convert them to chemically different forms. And the real curious feature is that after cutting a piece off of the 3' end, a new piece is put onto the 3' end by a special polymerase that does not require a template, but it actually adds a CCA to that end. I'm not sure why nature doesn't just code a CCA there, but it is clearly necessary to have that sequence there. And so another enzyme is going to have to come in and add that CCA as well. Just to show you, I keep talking about it, but here's that pseudouridine. You don't need to memorize the structure of it. It's actually, however, I would add, kind of interesting to chemists. If you're an organic chemist, the holy grail of like organic chemistry, of course, is making carbon-carbon bonds. And here we have an interesting enzyme that actually takes a carbon-nitrogen bond, rotates the base, and makes a carbon-carbon bond out of it. So a lot of organic chemists have spent a substantial amount of time trying to figure out how that happens. But you don't need to worry about any mechanism there. I want to show you some of the modified bases that are present. Again, you're not having to memorize any structures here, but these are just some of the base modifications that are necessary to have a functional tRNA. So you can see these are all added by enzymes, and they can all be quite dramatic compared to the actual bases. And if you don't have them, the tRNA is not going to work. And really, the most notable aspect of this is just what makes studying tRNA so complicated in the lab is you can get the right shape, but that doesn't mean you're going to have a viable tRNA. Anyway, I did want to talk about the splicing process because this is the one that does use kind of a weird mechanism. Here we're actually going to require some ATP, and that ATP is actually going to be for building a good leaving group. So in this case, the energy is actually required. This is not just for driving conformational changes. It's going to start essentially by actually cleaving the intron out. So it is going to be cleaved. It's going to leave that 2,3+6 phosphate intermediate up there. That will be gone eventually. But the real key here is that we're going to bring in the ATP, and we're going to use that once again effectively to adenylate the second strand, the strand that's on the 3' side of things. So this is a mechanism you've seen before, right? We're actually going to build a leaving group very similar to what we did, not identical, but very similar to what we did when we had the DNA ligase. And then we have the nucleophilic attack displacing the AMP leaving group, leaving the phosphodiester bond there. This is a very different mechanism than any nuclear splicing, and group 2 splicing, and certainly the group 1 splicing. But really the most significant features of this, again, are the similarities to the ligase mechanism that we've already seen, and the fact that the ATP is actually being used now to build leaving groups, right? We're actually have to put the phosphate there with one ATP and the AMP with the second ATP, so this is actually an energy-dependent process. Just a couple things. Just to show you, to be kind of parsimonious, both ribosomes from RNAs and eukaryotic, the RNAs that are there actually contain other sequences within them. So rather than make separate genes necessarily for the tRNAs in some cases, they're actually just tucked into the genes or the ribosomal RNAs, and those get processed out. That's interesting, but I don't know if it's the most important thing. I did want to tell you a little bit about this, which seems like a strange place to put it, but why studying some of this stuff actually can turn out to have some pretty significant importance. I'm actually showing you another immune receptor here, one we haven't talked about in this class before. This is a class of receptors, collectively known as toll receptors, and basically they just are good for sensing like gunk that doesn't belong inside of you. Just put it that way. Things like single-stranded RNA or pieces of bacteria, peptide glycan, all sorts of things you have these specialized receptors for on some of your cells. But you'll note that I also pointed out that there is one particular toll receptor on your immune cells that's there for recognizing RNA. Because what should you have floating around inside of you is a lot of free RNA. And if you do have a lot of free RNA floating around inside of you, that's a pretty good idea that you've been infected with a virus. And so some of that free RNA will bind to your toll receptor and trigger a really, really potent immune response, not specifically against who made the RNA. It's not like, oh, you made the RNA, so I'm going to kill that cell. Just one of those kind of things that drives your fever up, makes you sweat a lot, loses your appetite. Yeah, that's all because of your toll receptors, which, by the way, are really good, right? Because they help in keeping you alive. Because, again, most of the time when you have RNA inside of your body, that is mean just floating around, not the stuff maybe sequestered away inside of your cells. You've probably been hit by an RNA virus, and it's a pretty good idea to raise your temperature, maybe make you less hungry, all sorts of responses. But this toll receptor does create a problem. And the problem that this toll receptor creates, of course, is that not only can you not get RNA inside of you because of a virus, you also can't make an RNA vaccine against everything. Because for a long time, people realized that instead of spending all this time to either deal with pathogens and grow up lots of pathogens to make weakened pathogen vaccines, or making lots of proteins, which can be really, really time-consuming and an enormous effort, wouldn't it be a better idea to inject you with a little bit of the mRNA against the thing you want to fight off, let your own body make just enough of it, and then that would be a great vaccine, but it doesn't work because you have toll receptors, and that's the problem. And fortunately, Caitlin Carrico, work that I'm sure we've all experienced at this point, literally firsthand, figured out that you can make mRNA and replace some of those bases with some of those base modifications I showed you, and your ribosome will still read them as though they are viable. And it turns out that the toll receptors, because they never evolved to see these base modifications as dangerous, don't attack you. So it's not perfect, right? Because you can't make it with all base modifications, which is why you possibly, just like me, felt really kind of cruddy for a day or so after that mRNA vaccine that you might have gotten. But the key is they don't trigger a full-bore-on immune response. Anyone who had before tried and didn't understand base modifications or thought to try base modifications failed at the idea of making an mRNA vaccine because it was known your toll receptors are just going to like make you so insanely sick you're probably going to die from that level of RNA receptor to you. But Caitlin Carrico, who was not only familiar with toll receptors, but also the fact that, hey, there are these base modifications. We could try out a few of them, is able to take advantage of this knowledge from this PowerPoint, of course, and make these vaccines that really transform everything. So I kind of wanted to close our discussion of RNA on that. On Wednesday, we can start to use some of this great mRNA to see how we begin to make some proteins out of that. Okay, right, why everyone is here for?